Good afternoon, everybody. Welcome to the colloquium, the C3.ai Digital Transformation Institute weekly colloquium. Just to remind everybody, the mission of C3.ai's Digital Transformation Institute is to get together a group of scientists and engineers and all ilks to put together the science and technology of digital transformation. It has as partners certainly C3.ai and Microsoft, co-led by Illinois and Berkeley, with CMU, MIT, Princeton, University of Chicago, Stanford, Carnegie Mellon, and I said Carnegie Mellon, and NCSA at Illinois, and the Lawrence Berkeley Labs. We have a full calendar of events this week beyond today's lecture, and I'm going to introduce Teresa in a minute. But just to give you a preview of the talks to come, Alexander Madry, MIT, will talk about when machine learning models fail. I've seen a preview of this talk. I think it's fantastic. Richard Baranyuk from Rice will talk about insights. He's been working on better technologies for enhancing learning outcomes in schools. Lillian Ratliff, University of Washington, will talk about learning on how it is that when you introduce automation, people that you introduce it on don't stay stationary, but they actually change their behavior. Steph Bertozzi and Juan Pablo Reyes, they spoke also in our annual symposium last month. They'll talk about the state of the transmission and in Mexico of the SARS-CoV-2 spread in Mexico, and that also is really intriguing talk. Zika Korta, Carnegie Mellon, will talk about equilibrium approaches to deep learning, and Anna Horton and Jonathan Ozek from University of Chicago will talk about inequities in health. Certainly one of the things that's most in the papers nowadays in the studies that she's been doing. Actually, this is, we are told it's not yet public, but it's award-winning work. She's just been nominated for some wonderful award. Okay, and going forward, of course, there's additional wonderful speakers. We have an architect, Stefana Porosco, Christian Borgs talking about networks, network theory, one of the prime architects of network theory, and then deep learning through optimization, Cerebro, and mathematics of deep learning from Matis Stelgarsky. And finally, Stefano Soato, who's leading the deep learning efforts at Amazon, and also a professor at UCLA and Caltech, who will talk about differential learning. Two more talks. And just to let you know, the repository talks are really quite a wonderful library of events, and they're on the YouTube channel. You can hook up on the YouTube channel. It's under C3 Digital Transformation Institute. You can also go through our website. And I must say I enjoy looking at the talks from even early last year. They're just really wonderful. Okay, it is also my pleasant duty to inform you that the second call for proposals is again on a topic, a timely topic. It's about AI and machine learning for introducing energy and climate security. And the call is just out, AI for carbon sequestration. So these are the topics. Let me not read through them. The call is on the website. We'll have two webinars, one on the 15th, and there it is. The two information sessions, a webinar on the 15th, which Tandy Warno from University of Illinois will host, and one on Wednesday, February 17th, which Kostas Panos will host here at Berkeley between 11 and 12. And then there'll be additional computing resource webinars on the 19th, 23rd, and the 2nd. The proposals are, and also there's office hours. We really have a wonderful staff in C3.ai DTI that are available to help people with their computational efforts, and they'll be holding online office hours and so on and so forth. And that is the key date, March 29th. Proposals are due. And today's format, of course, a 40-minute presentation, 15-minute Q&A. Teresa is our speaker, and she will tag team with her postdoc scholar, Dr. Moitaba. Uh, I'll pronounce his name in a second. Moitaba, I'm sorry for, I don't want to screw it up entirely, but I will pronounce it correctly when we get to it. And it is about small molecule drug discovery using physics-inspired machine learning. Teresa is a colleague of mine, and she's a professor in bioengineering and chemistry. And she's a very distinguished teacher, but she works on really across a lot of different topics: molecular biology, nanotechnology, and advanced scientific computation, to really think about new ways of theoretical and computational chemistry, and which are exemplified really in this project. She has a abiding interest in the development of complex chemistry models, accelerated sampling methods, coarse graining, and multi-scale techniques. I've watched, not only watched the methods and models developed in her lab being disseminated, but she has built a consortium of some of the top universities in the country to pull together an academic program in how you disseminate these computational methods for chemistry and biochemistry and molecular biology. So it is indeed a great pleasure to welcome you, Teresa, and I will turn it over to you, and perhaps you can introduce Moitaba as well. Thank you.
Thank you. Thank you, Shankar, very much for the very nice presentation. And what I wanted to start off is to welcome everybody. Thank you for coming. As a theoretician and a computational scientist, I find that during the time of COVID, life has been has accelerated in research, and I actually have an obligation that I need to take care of, and so we're going to do a tag team effort, which I think is actually quite appropriate, with Mataba as has the lead postdoctoral scholar working on the project, but also to emphasize in our attempt to use machine learning for scoring drugs for COVID-related proteins, that it's a community, and the community involves researchers here in California at Scripps and UC San Diego and Berkeley, but it also involves national efforts, of which I'm a co-director in a project called MOLSI, an NSF-funded software institute that plays a role. And also that because of the importance of the COVID project, that it has expanded internationally. And one of our organizing hubs in Europe is BioExcel. And so I wanted to thank everybody, in case I get lost in the presentation and the science and forget to say thank you. I just wanted to say thank you to all of the team members and all of the collaborative efforts that I'm going to talk to you about today, to show you the community response in our community, the computational molecular science community, and how we've responded to COVID. Okay, next slide, please. So I wanted to, of course, remind us of this remarkable year that we've experienced, and the timeline of the disease. And up until late 2019, there were 4 known types of coronaviruses. These are showing you a model of it here. 2 of them are what we know cause common colds, but there are 2 that actually result in severe lung infections. And one we know of as SARS, severe acute respiratory syndrome, and the other one is the MERS, the Middle East respiratory syndrome. And it turned out that then in December 31st of 2019, this new coronavirus was actually reported in Wuhan, China. And based on rapid sequencing and aspects of other types of biochemical and biophysical information, the coronavirus bore stronger similarity to the SARS version that causes these severe lung infections and was then named SARS-CoV-2. And then in February, the World Health Organization then named the disease. In other words, the virus is SARS-CoV-2, but the disease from the coronavirus is called COVID-19. And these were unknown before this outbreak happened in Wuhan. And they, of course, were concentrated in China, but we're a global community, and not surprisingly, the disease rapidly spread. And so that by March of 2020, March 11th, 2020, the World Health Organization declared it a pandemic. And what we were doing, as we became aware of this rapidly evolving situation at the Molecular Sciences Software Institute, we felt that we had gained a lot of trust in the community to be able to engage the biomolecular simulation community on this problem. And you might ask why. Why is this particular community important? And it's for the reason that that's where the deep expertise in drug discovery is concentrated. And so I hope to give you a flavor of the kind of the science behind drug discovery. And I think you will get some of that, but the idea is that our community really could play a role here. Okay, next slide. Yep, okay. All right, so let's talk a little bit about the biology of the coronaviruses. So first of all, coronaviruses are actually quite big. They're about 125 nanometers, okay? Think about that, think about your orders of magnitude in diameter. They are one of the largest genomes of all the RNA viruses. And so when you think about this, this virus holistically, then there's going to be a necessity for coarse-grained and multi-scale models. And again, this is something that our community does very well. The second thing is that the coronavirus protein was rapidly determined to be more deadly. And in fact, here's what we're showing off to the left is a movie of the spike protein, as it's called. And the spike protein is involved in early stages at the molecular level, early stages of infection. And in fact, what it does is that it actually binds to a particular receptor called ACE2 on human cells. And in fact, SARS-CoV-2 spike protein had evolved into a type of a sequence that gave rise to molecular interactions that were 10 to 20 times more strong, that were stronger in that binding. And therefore, it was one of the reasons why it was dangerous. And it was that atomic insight and understanding of how to calculate binding energies that actually helped biologists understand why this was a particularly dangerous virus. And furthermore, they're complex, right? Which is that there were lots of ways to dealing with viruses that, for example, could knock out, its mechanisms for protecting itself. But this virus was too smart for that. So something like genomic proofreading, which is then you could then develop an antiviral, drug that could actually, make it ineffective through mutations, was just not going to be possible. This virus essentially had come up with its own proofreading mechanism and could make changes to any type of mutations. And so at this point, then you can see that there's going to be a necessity for all kinds of chemical models, which is that our existing drugs are unlikely to be successfully repurposed, and we should think about it very broadly to anything from small molecule to peptide to antibody treatments is sort of all of the ways that we have to start thinking about it. All right, and so what we wanted to do was to put our community into action, and and so therefore you needed, first of all, data. Okay, not surprisingly, a lot of information you get a lot of information from the drug discovery process through information about structure and furthermore dynamics, which is what I'm showing here. Now, what does the structure tells you? What the structure tells you is essentially where is that binding region and how can you block it with a drug? Okay, that's one piece. The kind of the bleeding edge in drug discovery is recognizing that these big macromolecules are dynamic. And if they're dynamic, that means that they're going to periodically expose new regions of their surfaces. These are called cryptic binding sites. And if you can actually generate trajectories of their time evolution of these fluctuations, you'll be able to enumerate where those surfaces are. And if you can enumerate where those surfaces are, those are new drug target sites. And so what we needed then was a data repository. And so this is showing a movie, as I said, from D.E. Shaw's group, that using their high performance Anton 3 computer with their good Desmond software was able to generate the kind of the time scales that we need, okay, to see those fluctuations on the sort of millisecond time scale. But that's a lot of data. And so you needed a place to deposit it. And you also needed a place for it to get available to the community. And so MolC became an immediate clearinghouse for that kind of data. But you also need an intellectual repository, which is that we have a lot of smart chemists and physicists and biophysicists, biochemists. We have all kinds of material scientists. All of these are really smart people, but they didn't know much, let's say, about virus biology. And so it became then also a resource for education that would let researchers get going really quickly. It would tell you about the biology. It would tell you about what were the knowledge gaps, what were we missing, and therefore, what could be the computational tools that you could use to get to the state of the art really quickly. And that was the goal. That was a way to essentially get the whole community involved by letting them, get that background knowledge really quickly. Okay, next slide, please. All right, so MolC then also had established some trust in the community. And I think that we all know that science is competitive, right? It's a contact sport. And, we're all naturally pretty competitive. But if you're going to tackle a genuine crisis, where your community can play a big role, you've got to enable cooperation. And so, the combination of MolC and other community leaders like Rami Amaro, who I'm showing here, who's my co-PI on our Data Transformation Institute grant at UC San Diego, and then our international collaborator, Adrian Mulholland from the UK, quickly put together a big community letter that I think had on the order of around 250 or so signatures to emphasize that we would cooperate on data, and that we would cooperate on intellectually on trying to really work on this problem in advancing drug discovery for COVID-19. Okay, so next slide. All right, so we kind of got down to work. And so what we did is we had some young molecular scientists that were ready to roll. And so on March 28th, our board, of which I'm one of the co-directors, convened, and we took resources away from current projects, and we deployed these software scientists that I'm showing here off to the left. Levi on the top there, Naden, and Sam Ellis, and Andrew Abi Mansour, and Ben Pritchard, and Doa Altawari, who had lots of expertise in everything ranging from molecular simulation to quantum chemistry to data science and machine learning and workflows and all of those good things that needed to come together. All right. It turned out that we got organized so quickly that the big consortium, which is much bigger, honestly, than MULSI is, because it covers all of Europe, kind of took notice of our efforts, and they just decided, like, look, okay, let's just join your guys because you guys are so far ahead. And so we then teamed up with our European colleagues, and that's how we became a co-consortium. And then our mission was initially just to start finding ways to bring that data together. And so we created this centralized, curated repository and trajectories, and also started to make available what was originally proprietary algorithms and software, which is that we had a number of companies who came in who agreed to give us some access, okay, to their software that you originally had to purchase in order to make progress on the problem. And so again, so we set up this initial stage, and I had that going by the end of March. Next slide. And then what we wanted to do was to inspire the young ones out there who really wanted to be able to participate. And so what we did was we put together a special software fellowship program. And what this allowed us to do was to bring in, with extra money, we set aside extra money to bring in 5 postdocs and students who could work on, the COVID biology with their techniques. And so this went through rapid peer review and gave rise to then the seeding of these young researchers, getting them involved in obviously an extremely important problem for the world. Next slide. Okay. So I wanted to give you a little bit of sense of the things that they were proposing, which is that some were developing things like workflows. And what they were doing is they were trying to generate novel and recognized novel therapeutics for the SARS-CoV-2 virus. And so there's postdoc James Krieger. We had second-year graduate students, who were developing ways to model those cryptic binding sites, right? Those conformational changes, okay, on those big proteins requiring much more efficiency because of the size, as I've already sort of indicated. This is a big problem, big system. And then we had deep learning approaches, of course, because there's of the data, and this came from the folding at home part of our community, and others who came in with computer science backgrounds who were working on things like antibody design and other approaches. So the idea is that it really ran the full gamut of the kind of methods and theoretical frameworks and software that one would need to tackle the problem. Okay, next. All right, so then we started working pretty hard, and we, on the week of March 30th, a lot of stuff started coming together, which is that we established a lot of resources for the community: high throughput computing, HPC resources, cloud resources, which quickly got established, that became available. This repository of actually now populating it, having set it up and in a logical way, now the repository came in, and so we reached out to the international community. D.E. Shaw's group, shown here with Brian Jackson, they made available that very high quality trajectory data on that spike protein. The Sugitas, Yuji Sugitas and Makoto Taji, who are in Japan, had actually gotten special permission to actually generate simulations before their new computer, their new high performance computer, actually became available. And so, so Riken then was able to then give us this protease data by mirroring. We began then talking to the European community, and under the CERN umbrella, right, where of course those are dealing with particle physics, where the amount of data that they generate is enormous. They were the experts, right, on how to handle data, and really, honestly, our data at this point, or at then, was puny by their standards, but they were very eager to help, and therefore gave us, expanded our little trivial 4 terabytes, at the central physical place of MolC, to now essentially giving us access to real data resources with working with Sonato, which was under the CERN umbrella. And as I said, which is that the community letter got published and things were underway. Next slide. Okay, so then what we wanted to do was to kind of recognize a few things going on here, right? Which is that if you're going to bring all this data in, and nothing's published at this point, and so how should you view this data? You got to be careful with data because garbage in, garbage out kind of problems. So what we needed was a consortium of people who could actually evaluate the data that was coming in, and to also give evidence of where it stood in regards to validation. So we created this, stoplight system of red, yellow, and green with experts that I'm showing here as the community began expanding to make sure that we could trust that data. And furthermore, to really talk about the biology, which is that to make the connection between the molecular questions of drug discovery and how that could connect to the COVID-19 biology. We needed experimentalists, okay, that were generating not just structural data, but bioassay data in regards to whether repurposed drugs at that time were going to be effective. And as I said, the potential therapeutics could be small molecules, they could be peptides, they could be antibodies, and of course the vaccines. And so some of the effort of those vaccines always has a modeling step, and some of the people in this community were involved in that process. And then, okay, so I think that then gives you this idea that then, like I said, the key thing is that there's this fear that there was going to be a generation of bad data, and we wanted to avoid that. So there was this ethical responsibility to make sure that the data that was coming in was solid. Okay, next slide. Okay, so we started to really, as I said, continue to expand, and we really were now starting to really get a flood of data coming in. We started getting sort of genome-wide data. We were getting whole virus models. We were getting lots of new drug molecules that were coming in from different segments of the community. And at the same time, the community needed to somehow get some way to document, okay, what they were doing, and so we sort of started minting DOIs. We began negotiating with the European Bioinformatics Institute on how we were going to move off of these short-term storage arrangements to something that was going to be maintained over longer term, which is where the EBI made most sense. And as I sort of already had hinted at, we had a lot of really good cooperation from industry, which is everything from Schrödinger, which is a drug discovery company, through to NVIDIA, that were providing GPU accelerated resources for some of the tools that you need in drug discovery. And then starting to link, the fact that there were lots of data efforts, there's lots of different drug databases, and they're dispersed, and they needed to be there had to be a clearinghouse to essentially where to go look for that data. And so therefore these links started getting established as this community effort continued to grow. Next slide. And then what happened is that really, could you, one more time? There we go, there we go. So then what happened is that we really started to hit, data coming in from around the world. And so what we're showing here is a, world map of where all of this data was either coming in or going out, okay? And we started to also connect to a lot of, of larger collaborations that were, for example, working with AWS for, for our data sets. And we worked with Microsoft's AI for Good team, okay, which is, part of something that, we can connect here as well. Human Brain Initiative. It turned out that Human Brain Initiative thought a lot about how to think and about data, how to link data, just the way our brains work, right? And so it turned out that they had a lot of sort of ways to sort of think about data, how to query data, how to, to analyze data in new ways. And so they really wanted to work with us in the kind of data that we had collected. And as I said, we were connecting to, to JEDI and the Open Science Grid. I mean, there were just lots and lots of organizations. And then this began picking up, in the government and the popular press, so HBC Wire, the two senators who were, because again, the physical site of the Molecular Sciences Software Institute is in Virginia. So those senators were, very pleased. Physics Today, was wrote what I thought, and I kind of wanted to sort of emphasize here, which is that how much everybody wanted to be involved. And so this article was about, how people were pivoting away from their kind of standard research days to try to engage on this, on the and trying to find solutions to this world pandemic and this truly altering of our lives that we've experienced. And when you're working on basic research, which is where I'm at, I kind of know that I stand back in awe of the fact of how this virus has changed our lives in so many ways, and that but at the same time, we're starting to head out of it, right? Which is that a vaccine and several vaccines have come into play, and various types of therapeutics. And in fact, I'm actually going to get vaccinated tomorrow, and I can't believe it. And so, we're going to be coming out of this, but I think that what we've done so far is that we've built the capability for next time. And so I think that's what's really the result here, is that we know there's going to be a next time, and that's what we need to be ready for. And I think that we will be. I think that we got this together very quickly. And okay, so anyway, so that's sort of some of the background of how the data and the organization happened. Okay, so now to the next slide. So now I'm going to talk about the fact that even though a vaccine is coming, there are, we have a lot of political resistance to the vaccine. There's still a need, right, for therapeutics if you get COVID, right? And one of the areas where we're working on with Rami Amaro and also Stefano Forlì at Scripps in San Diego is to be able to score drugs, so to essentially find new drugs. And it looks like the repurposing of drugs is probably not going to, going to carry us as far as we like. And so the thing is, what is holding up the discovery of new drugs? Well, there's two things that you can think of. One is the exploration of chemical space, which is that, given, there's certain drugs, small molecule drugs have to be a certain size, they've got to be bioavailable, they've got to have a number of properties. And so when you kind of add up all the features that are necessary to be a plausible drug, it's a large chemical space of something like 10 to the 64 possibilities. And so how do you know, therefore, whether they're going to be a good drug? Are they going to target the right protein? Are they going to target the right region of that protein? And therefore, are they going to be selective? And are they going to be tight binders? How are they going to be delivered? All of those things means that they have to be scored. And the most basic thing you got to do is you got to get the drug to bind. And to do that, you need to come back down to then a way to score it. And the most rigorous way is just H psi equals E psi, right? If you can solve H psi equals E psi, you're going to do it correctly. Well, why can't we just solve H psi equals E psi? Because it's expensive, right? Which is that LeConte in the 20s, said that, we have now discovered all of chemistry, all of biochemistry, and most of chemistry and physics with understanding H psi equals E psi, but the equations are too complicated for us to be able to solve them. And so what over time our community has developed are very robust approximations. Those approximations are really good, but even so, they're still too expensive for doing something like high throughput virtual screening, which is what you want to do, which is you got to target and you got to be able to say, and you got a drug and you want to ask, is that a good binder? And so what we're doing in this project is that this is the weak link in doing high throughput virtual screening. This is the gap. And the gap is that you can dock drug molecules into pockets on a protein. But as far as its ranking, whether that's a good doc score, it's a little bit all over the place. And so that's what we're trying to bring to the project is we're going to bring high performance computing, machine learning, and we're going to put all those things together by raising the physics to be able to score these drugs, not one at a time, but in a high throughput way, okay, for virtual screening. Okay, next slide. All right. So what I'm going to do is that I apologize again. I have to head off to my obligation, and what I'm going to do is put you into the hands of Dr. Hagliardi, who is going to tell us about how we then take those scores and use them in physics-based machine learning. Okay, so thank you very much, everyone.
Okay, hello everyone. Sorry for the interruption. I get to the job as quickly as possible to just continue this presentation. So far we discussed that there is a need, of course, because of the challenge that we have currently in the world to find new drug molecules and target the COVID-19 proteins. And for that, the first question is that what's the space of the drug molecules or those so-called molecular space that we have to explore. And of course, the immediate question after that is why bother generating new molecules? So first, I have to tell you that the molecular space is estimated to be a size of 10 to the 60s, and actually, of course, it's infinite because if we consider all elements in the periodic table and then try to combine them based on the chemical rules, we can get an infinite number of these molecules, but we can also limit them based on some criteria that are some rule of thumbs, like Lipinski's rule based on their weights or for example, the hydrogen bond that they can create. So the size can be limited, but it's estimated to 10 to the 60s, which is a huge space of the molecules. And then we haven't been, I have to acknowledge that we haven't been the first group who is attempting to explore this molecular space. So there have been a long history of the methods to generate the library of the molecules, for example, based on the combinatorial library generation. And in this table, you can see a number of those data sets that are well known, and there are many data sets that are just organic molecules, small organic molecules that have been generated for different several applications. So the main goal here for the combinatorial library generation is to start with a pool of building blocks of the molecule, some moieties that are known or based on the feedback of the experimentalists, they are promising for some applications. And then based on the reaction based combination of those molecules, we can enumerate the space. But the problem here is that what we get at the end is a very discretized space. And the other point that we have to mention here also is that those targets that we get in the molecular library space, that the molecular libraries basically is not necessarily those good candidates that we need to target the COVID-19 targets. What we have to do now is going after the de novo molecular design. That is also is a very recent effort in the design of the molecules. I need to go to the next slide. Yes. And for the molecular design, now I have to mention and also give you an overview of the methods that have been already developed. So we are not at the beginning. We are not going to reinvent the wheel. So this is already, some of these methods are already developed. Some of them are shining like little stars, but the others are the other methodologies are basically buried behind those that are mainly advertised. So we need a consistent profiling of the methods, and we need to find out what methods we need to take as soon as possible to address this challenge that we currently have quickly. So for the molecular design, the first thing is the representation that you see on the top left corner. So for the representation is basically how we want to represent or how we want to describe molecules to machine. If we want to explore this molecular space using the machine learning methods and data mining tools. So for that, I have to mention that we know that for that we have certain dimensions of the molecules to be described, or like descriptors of the molecules. The first dimension is the atom type. So you can consider, I'm sure everyone here at least has had an organic chemistry course in high school. So you know molecules look like graphs, like we call it molecular graphs that have nodes and edges. Nodes are atoms, edges are bonds. And then based on that, if we have just information about atom types, that's the first dimension of the descriptors. And then if we have information about how they are connected to each other, that's the connectivity, or the second dimension of the representations. And of course, the third dimension here is based on the spatial coordinates and the relative distances, to consider all interatomic interactions, not just those that are bonded to each other. So based on that, people have been developing methods to also have mainly, as I said, machine understandable, but it's better to say it's like a numerical representation for the machine. So what the point here for the molecular design that I have to emphasize is the fact that for this particular problem of generating new molecules and also exploring it, biasing the distribution, we have seen that the very raw inputs based on the strings or those encoded text that we call it SMILE representation, or more recently a new approach called selfies developed in Alana Sperguzic group, so have been really promising, although they are not, there is not lots of the handcrafting and adding more or extracting more information. It's very, very similar to what we do in the natural language processing, mostly, I mean, in the computer science community. I'll show you an example of this representation of the molecules and how they can be used in this generative task. On the machine learning methods, which might be more interesting to this group of audience, also there has been very interesting efforts that you can see in this review paper published by Sanchez, Benjamin Sanchez et al. So you see that many of the generative methods that have been already developed in the CSC community has been used, and some of them have advantages, pros and cons compared to each other, to the other ones. So the first one is variational autoencoder, that the goal is to get a molecule that you see here and then encode it to a latent space, but instead of having just 1.1 representation of the latent space, similar to what we do in the autoencoders, we want to have like a latent a distribution of that latent space, which is the points that are important here are continuity of that space and completeness of the space, because we want to be able to sample from that space and then feed it back to the decoder to generate the same molecule based on the input representation that we picked, and be able to reproduce that, reconstruct that. If we are able to do that, then later we can use this distribution and bias it towards the areas that can provide more interesting, favorable targets, those drug molecules that can target the COVID-19 proteins. So there have been also other methodologies. For example, GANs can be adapted to this problem if we start with random noise and then having a generator to generate something like molecules, but the main part is to have a discriminator to also feed in those molecules that are valid based on the physical rules, and then try to just classify it as fake or real ones. So at the beginning, the real ones are those that we are providing as extra information, or extra, like those valid molecules that we are feeding in, and those that we are generating won't be really valid. And down the road, it starts to tune the parameters in the generator also to generate more valid structures. So there have been Monte Carlo research to go to an end point of a branch of the trees and provide the feedback to take those trees that are generating valid structures. And then the other one that I want to really focus on is the recurrent neural networks. And this is, again, very similar to the approaches in the computer science community, mainly for the what we do, for example, what we use daily in our Gmail text writings, when we are writing an email and we have an auto completion or some suggestions based on that. So this is based on a probability distribution over the next tokens of the sequence that will be provided by the model. And the model is already trained on a bunch of these, like a training data of these sequences, to know how to provide such probability distributions. And here you see an example of a smiles that I promised to show you. For example, these are just letters. For example, C refers to carbon, but there are also other letters that don't directly point to like atom types. For example, this index 1 that is used to close a loop or like a ring in the molecules, or other symbols here that, for example, show a double bond or a triple bond, something like this. Okay, so here is a review of the methods. So as I said, we are not going to reinvent the wheel. We are going to take the best approaches, but at the same time, based on the efforts that we have to infuse the physics to the model and also improve the scoring, we are developing new ideas, and mainly to address the challenges that are involved in this problem, and also to accelerate the screening, as Professor Hedguru mentioned, to have an accelerated high throughput screening. Two more important, or let's say all of them are equally important, but two other approaches that are really a main part of this task are first the choice of the metrics, and also the reward function. When we want to not only generate unbiased molecules, but also we want to bias the distribution of the generative task to towards the task that the target that we have. So for that, the metrics here, I have to mention, I have an honorary mention of the Guacamole. Is a Python library. This has been developed. It's a joint collaboration from the community to have this consistent profiling of the methods and providing the benchmarks to compare different generative models. And based on that, what is recommended is a benchmarking based on the distribution of the generative models, and also benchmarking based on this, they call it goal-directed, that relies on the target properties that we are working with. For example, here I'm mentioning QED, which stands for the quantitative estimated drug likeness, which is a measure of the drug likeness of the molecules. This can be based on some rules of thumb that are also mentioned at the beginning, like Lipinski's rule of 5, based on the hydrogen bonds, or based on the molecular weights and information like that, which are very efficient to calculate. It's not they can be estimated with no overhead. We can have log P or synthetic accessibility. For example, if we want to generate molecule in our computers, but is it possible really to also synthesize those at the end? Or the other thing is that if that molecule will be stable or not, or you can raise many questions. For example, if there is this molecule that we are going to generate is going to be toxic or not. So for those, we can have empirical models or data-driven models that can be helpful. And then for the choice of the reward functions, also we can evaluate this generative task and also the reinforcement task by by checking the validity, novelty, diversity of the molecules, and also taking care of the distribution that we are interested in, if it's like a biased and skewed distribution towards the favorable properties. Okay, this was a quick overview of all these methods. I hope it wasn't that overwhelming, but the current state of the art, also I have to mention that the people have been working on these different methods. One work that is really interesting here is this work that you see here by Gadibati et al. So what they do is that they not only try to generate molecules, but what they consider in the process of the generation is to have a reaction scheme and make sure the building blocks or the molecules that are going to generate the pool of molecules or the library of molecules can react to each other. So they kind of assure that what we get at the end is synthetically feasible. And based on that reaction scheme they are generating, and then later what they do is the approach of reinforcement learning to, for example, for the target property of PIC50, which is a very well-known property for the drug discovery. And here they target the HIV proteins for that. So you see that the orange distribution is a distribution that is biased towards the higher values of PIC50 after doing the reinforcement learning along with the genetic task. And here we also have QED, as I mentioned, the drug likeness or the log P, other metrics that is recommended to quickly screen the molecules and find those that are better candidates, and then later try to validate it using the experimental data and other metrics like toxicity or stability. So what we have been doing on the machine learning side, along with the scoring, QM, and all those methods that we are bringing into this task, is to have a generative model based on the AWD, LSTM. These are again recurrent neural networks that we feed in selfies. Is a similar representation to a SMARS, but by like the advantage of addressing the ambiguities in the SMARS representation in terms of the generative task. And then once we have that, we can generate new tokens. A probability distribution over new tokens can be used. And then every generative model should have a stochastic part, right? So the stochasticity here is that once we get the probability over the next tokens that we are going to generate to represent the molecules, we sample that randomly, and then feed into the model to generate the next tokens. And with that, we were able to generate many valid molecules, and then using a policy gradient method, we are able to not only give a feedback to the generative model based on the rewards that I mentioned based on the physical properties, but also based on the validity of the molecules again, because we don't want to perturb the generative model and keep it to generate good structures for during the iterations. So with that, we have been able to improve the QED, as you can see here, by iterations of the joint training of the generative model and reinforcement learning. And then you see that the distribution have been already shifted. So we are using this now as like a protocol, a workflow that we have based on the state-of-the-art methods to quickly adopt it and apply it to the methods that we are also developing on the scoring side. And then down the road, based on the validations with the experimental data, we will be able to find the top candidates, hopefully, that can be good drug molecules for this disease. This is my last slide. I just wanted, again, Professor Head Gordon mentioned at the beginning, we are really grateful, thankful to the C3AI for the support. And I also wanted to thank the informatics team and the support that we received in terms of the resources. They have been super accommodating to provide all those GPUs, CPUs that we need, also access to the Microsoft Azure. So that has been a great work in at least past 6 months, and we are looking forward to the next few months to quickly address this challenge, hopefully. And of course, this has been a collaborative project with the entire team of postdocs students, not only in our group, but also in Professor Amoro's group in San Diego, UC San Diego. And thank you, of course, thank you all for your attention, and I'll be glad to take any possible questions.
Thank you all very much. I think that on the chat, there is a question about, may have been early in the talk, about that with underlying health issues included in the analysis. And
underlying health issues, I think it refers to the problems that we currently have. For example, different variants of the protein, the COVID-19 protein. I'm not sure about that, but if it refers to that, and based on what I just explained, so what we are considering is mainly those methods that can be quickly addressable using the computational methods, and can be also scaled up for a high throughput screening approach. Yes, the trajectories of these new variants of the proteins should be collected, but I'm not the best person to answer that if it's going to come soon or we have to wait for such information.
let me ask you a question. So, you said towards the end of your talk that you're using these GANs and all these other methods. And what is your experience about, there are, as you know, the speaker next week in the series is going to be talking about his concerns about the use of GAN. And so what is the view from the point of a user about how well these methods are working for you?
So we have this benchmarking. So I have to tell you that we are not particularly glad about GANs. So we mainly work with the recurrent neural networks or variational autoencoders. So based on the benchmarkings that we had, and also for not just this project, we have been using these methods for other applications. And yeah, so
let me rephrase the question. Okay, so I think that the community feels that these are wonderful sets of tools. There are Python libraries, and you mentioned some of them. But from a user perspective, we're also hearing from groups of people that the algorithms are brittle, they're sensitive to training data, and so I wanted sort of for different flavors. It doesn't have to be GANs, RNNs, CNNs, whatever. So I just wanted to get your own experiences about using these for your your scoring drugs. So I think the scoring, a lot of the applications which are classification, they do well. But scoring is a more analog, it's a more qualitative thing. So that's why I'm curious as to what your experience is.
Actually, this is a very good question, and I want to tell you my personal opinion about this. That I believe in this task, the key, or like I can't emphasize more, that the key point is the scoring functions. That we have to have very physical scoring functions that are reliable. For the generative task, even if we are getting a validity of 60%, 70%, we don't care. I mean, we should care. But the point is that, okay, if we are not generating good molecules or valid molecules, we toss them. And we keep those that are valid. And hopefully and we have seen that doesn't really affect the distribution that we are getting, because we are working with a continuous distribution because of the size of molecular space. So you know what I mean based on the we don't care. I mean, basically, let me to explain that this has been current concerns in the community to make sure we are having 100% validity. Although we can reach to that point using, for example, recurrent models, but the point is that even if we don't have such validity, it's still good. Because the generative task alone is not really the part that we care about in the physical domain, or I mean, the scoring and validation of those drug molecules are super important.
Very interesting. Very interesting. Okay, the other, I was just waiting to see if there are other questions. I don't want to hog the question, but I ask one more question. The people using the AI methods in the COVID in this have complained that because they need huge amounts of training data, and the data is there isn't so much data. There is data, okay, there's lots of it, as you said, but maybe not so much in an absolute sense because of the new disease and so on. But you have not felt that your algorithms are data starved. You found that there's enough, or is that why you gave me this more nuanced answer about 60 to 70%? Where you're just doing a simple classification. What is your? I'm just probing to understand how the methods are doing still.
Yeah, so first of all, about the data, yes, there are already some available data sets, but we don't rely on those only. That's why we are collaborating with the experimentalists also to get a sense of the scoring functions that we are using. And also we are generating the QM data, quantum mechanics data, that can be considered as a gold standard for the computational studies. I mean, not comparable to the experimental for sure, but at least for the computational data sets, this is a good data set. But also for the generative models, yeah. So currently with the recurrent neural networks, we are able to get higher than 95% validity, and so ignore my comment on the fact that we don't care, but yeah, the validity is good. But I mean, just generally the discussion that is going on in the community is like this.
Just tune in and listen to next week's talk. The Alexander Madry, he'll talk about sort of the sensitivity of the networks to small changes in training data, and that's his concern. And but anyhow, if it's doing well for you, that's great. Okay, I think they're already past the hour, so I want to thank you very much. That was a very interesting talk, and I have to say I want to plug your consortium. I was involved with talking to Teresa, who I think had to disappear, in setting up your consortium. So it's really wonderful to see this consortium really step up to this challenge of And so congratulations on being the lead artist. Also, just to mirror Camille, you know, the talks are going to appear pretty PDQ on the YouTube channel. She's saying by next week, but I think we might be sooner than that. And I hope we have your permission to post the you know, your talk in some fashion. And I think either Larry or Camille will talk to you about this, Mataba. And okay, I don't know. By the way, we were. I don't know if it has begun yet. Oh, Teresa here. That you know, we were going to take somebody. We're going to take the help of a automatic transcription machine. It may not be. May not have been activated in time for your talk today, but if it is, you'll get a transcript of your talk as well.
Okay.
So
Looking forward.
Thank you very much.
Thank you so much for the opportunity. Thanks a lot.
Wonderful.